Microbiology is one of the broader fields that exists because basically it's the biology of microorganisms. So you can be an ecologist, you can be a molecular biologist, you could be an evolutionary biologist and still study microbiology. My particular area of interest is detection of pathogens in the environment. So I usually sit down at the computer, answer some emails, check to see if any students have any questions or want an appointment for the day. And then I usually prepare for lectures or labs and then go to those activities. So we have contact hours in terms of the time we're actually in classroom, which is around 15 to 18 hours a week. And then the rest of the time we're either preparing for those classes or we have other types of meetings on campus or meeting with students. Plant physiology is? So plant physiology is a study of basically all of the functions of plants. So from anything from how do they adapt to a given environment to how do they carry out metabolism, but it's any function that the plant can do. I was interested in plant nutrition in terms of, in particularly in what is called subsistence agriculture, where basically people don't have the ability to add lots of fertilizers and things to grow their plants, and so how can you increase the plant nutrition? And so that sort of was the kind of thing that got me into plant biology. I wanted to teach more than I wanted to conduct research, and Carroll College is a teacher's college. Plus living in Montana isn't half bad. In terms of, in the current lab, the students are trying to identify organisms that I've given them, right, that bacteria, that they don't know what they are. So they're doing a number of physiological tests to try and identify the organism.
Okay, thank you.
Yep, thank you.
Questions? Oh, yes, I'll try to answer them. Okay.
So what do you guys do?
He gave us an unknown organism. And here, see the little white dots on top growing on the auger? That's what we're right now. And we have to burn this to make sure there's no organisms on this, because we don't want to contaminate it and get all the other ones in here. And so you burn it and you put some organisms on here, the unknown, and then you spread it out in a pattern. Can you see the pattern on there? A little bit. And then that makes it so it grows in little tiny colonies instead of all.
And we do that for a variety of media. So we have some
Thank
mannitol salt.
you.
Oh! Sure. Well, what we're doing here is we have a variety of different kinds of what we call agar. So basically bacteria, they need nutrients to survive. And so what we do is we provide nutrients, but in different forms. And so we have 3 different types of nutrients they can eat off of. And we use that to kind of figure out who they are, because certain bacteria like certain nutrients and other bacteria don't. So by combining all the different ways that they survive on these different media, we can figure out who they are. And sometimes what's cool is not just them being there, but sometimes, for example, on this is mannitol salt agar. And if it's able to ferment mannitol salt agar, you'll see a color change. So not only will you know that it can survive on mannitol salt, but that it's able to ferment it. And then you'll see it change to, I can't remember, is it green? Greenish color. And so that kind of is another indicator of what it might be. Oh, you're welcome.
Alright, you will have 2 or 3 knowns. So if you have 2, just split your plate in half. If you have 3, divide it up into thirds. Alright? All you need to do is inoculate a dime-sized spot. Alright? So you'll have 1 plate of everything for your knowns, and then you will have 1 plate for your unknowns. This is per group, not per person. Alright? And so, if you're in a group of 3 again, like you guys, you need to divide it into thirds. If you're in a group of 2, just divide it in half.
Do you have any nets in?
I was looking for that.
Yeah, it looks like you got some back there. Okay. So, that's Nile stuff. Now, do any of you guys know why how mosquitoes find people or animals that they're going to bite?
Oh, wait, no.
What they use is they can tell when we breathe out, we let out CO2, and they can sense that. So what we do is we trick the mosquitoes. We don't have a tank in here that I can see, but these are kind of gauges that we hook up to CO2 tanks. So we have these big, like, scuba tanks filled with CO2, and what we do is we let just a little bit of the CO2 out of these little tubes, and then we attach them to these traps over here. So
how this works, we got our little collection container here. We put a net over it. And we screw that on with this little top. Once you get that on there, you hang them from a fan. You can see that little fan in there. They hang this top part from the fan. And the mosquitoes can fly into the top there.
well, I might be able to see it better.
See how there's like mesh in there? They can fly in through there because they smell the CO2 and they're like,
Sucks them
ha,
in.
people I can bite. But instead, the fan sucks them in and they get trapped inside these little nets and they just collect in the bottom. And you can see right through there.
What happens when you take them out?
Well, we don't want them to start. A lot of them will be dead, but not all of them will be dead.
We don't want
So
them to
first
get
of all,
squished.
we want them dead, and we want to make sure that the virus doesn't degrade, because after time, the virus can actually just kind of die on its own. So what we do is we put it on ice. See those giant chests over there? We fill those with ice packs, and then we try and keep them as cold as possible. That preserves the mosquitoes, and it prevents the virus from being degraded.
So that's